We'll start from mitochondria. Can you tell me what is the function of mitochondria?
Um, Aerobic respiration.
Okay, and it's also known as the powerhouse of the cell. The structure of the mitochondrion, as seen with the electron microscope, is visible in different pictures that we'll see. They are usually 1 micrometer in diameter and can be various shapes. Sometimes the sausage shape hoti hai iski, something like this. This is what a mitochondria looks like. And inside we have these different envelopes that I told you last time as well, which was cristae that are projecting into the interior of the mitochondrion. And the cytoplasm of mitochondria is known as the matrix. Now, the number of mitochondria in a cell is variable. Why? Why do you think that some organisms have more mitochondria, some have less? What's the difference in that?
Because some need more energy for respiration because they like contract more, like muscles, or like they need more energy.
Yeah, exactly, Chike, because they're all different. Some need more energy, some need less energy. And as they are responsible for aerobic respiration, it is not surprising that cells with a high demand for energy, such as liver and muscle cells, contain large number of mitochondria. A liver cell may contain as many as 2000 mitochondria. And if you exercise regularly, your muscles will obviously make more mitochondria as well. So the main function was aerobic respiration. Any idea what was the equation for aerobic respiration that you studied in IGCSE?
Uh, Oxygen plus the glucose produces carbon dioxide and water.
Good. So, although they have other functions as well now, this is something new. Synthesis of lipids. A series of reactions take place in which energy is released from energy-rich molecules such as sugars and fats, and most of the energy is transferred to ATP, adenosine triphosphate. This is the energy carrying molecule found in all living cells, and it is known as the universal energy carrier. Christie, that we have seen, the inner membrane folds of mitochondria envelope, they have these stocked particles of ATP synthase and electron transport chain associated with aerobic respiration. Adenosine triphosphate, the molecule that is the universal energy carrier. ATP is like a battery. A battery contains energy. This ATP molecule basically contains energy because energy cannot be, transferred in completely raw form. It has to be a part of something. It has to be transported in some form, which is adenosine triphosphate. Now, the reactions of respiration take place in solution inside the matrix and the inner membrane, and this is all from chapter 12 of your A2. In A2, chapter number 12, you'll see that how these respiration different stages, there are a total of 4 stages of respiration that actually take place in mitochondria. Now the matrix contains enzymes in solution, including those of the Krebs cycle as well. Then there are some electron carriers as well. You just have to remember the structure functions you'll study in your A2. Moreover, how is energy given out then? We know that ATP carries energy, but can you tell me how is energy given out from the ATP? Energy is made, converted to ATP. Now, how is ATP gonna give us energy?
Uh,
It is going to break down into adenosine diphosphate and phosphate. What does triphosphate mean? 3-phosphate. It is going to hydrolyze into diphosphate and phosphate. This reaction basically produces energy that gives us energy. Got it?
yeah.
If we need more energy, this ADP would be converted into adenosine monophosphate if we need even more energy. So as you remove one phosphate, another phosphate, energy keeps, spreading around the body. Because once you make energy from aerobic respiration, that energy is converted to ATP. Now this ATP molecule is spread around the whole body wherever there is a need of energy. And this ATP has to, dismantle as well. It has to give out energy as well. If we loaded energy into this ATP, it has to unload the energy somewhere as well. It's a transport. Got it?
Yeah.
Now, what is the endosymbiont theory? This is very important. Okay? It was discovered that mitochondria and chloroplasts contain ribosomes, which are slightly smaller than those in the cytoplasm, and they are the same size as those found in bacteria. Cytoplasmic ribosomes are ATS, which are those of bacteria. Ab ribosomes, I told you there are two types. What are those two types?
70F and 70F.
ATS. ठीक है? We know that ATS ribosomes are present in eukaryotes. 70S ribosomes are present in prokaryotes. But the funny thing is that actually mitochondria and chloroplasts also have ribosomes, and they are what? 70S. And the thing is that mitochondria and chloroplasts are also in ATS organisms. Then why don't they have ATS? Because this endosymbiont theory is going to tell you that these mitochondria and chloroplasts basically evolved from prokaryotes, because prokaryotes also have 70S ribosomes. Got it?
Yeah.
It was discovered in 1960 that the mitochondria and chloroplast contain small circular DNA molecules, also like those found in bacteria. It was later proved that mitochondria and chloroplast are in effect ancient bacteria. They are actually ancient bacteria. They were a part of those prokaryotic bacteria which lived inside the largest cells of animals and plants. This is known as the endosymbiont theory. So can you tell me what have you understood from endosymbiont theory? Hmm.
That mitochondria and chloroplasts evolved from prokaryotes
And how do we know that they actually evolved from prokaryotes?
because they have 70S ribosome.
Exactly. Because prokaryotes, overall, all prokaryotes have 70S ribosomes. Eukaryotic cells also have 80S ribosome, but eukaryotic cells organelles contain 70S, for example, chloroplast and mitochondria. Even though they are part of eukaryote, but they contain the ribosome which are 70S, which are a part of prokaryotes. So that means that they were evolved. Like we say, we have many, many qualities like the apes, monkeys. What do we say? We have actually evolved from apes. Don't we say that we have evolved from Homo sapiens? Because we still have some of the characteristics that were from the ancient times as well. That is how these two organelles, which are mitochondria and chloroplast, still have those characteristics from the ancient time prokaryotes as well. Got it?
Yeah.
Good. Endo means inside and symbiont is an organism. So endosymbiont theory tells us what is about inside the organism, inside the cell. We have gone through the organelle. The DNA and ribosomes of mitochondria and chloroplast are still active and responsible for the coding and synthesis of certain vital protein. But mitochondria and chloroplast can no longer live independently, and mitochondrial ribosomes are also visible as dark orange dots. Now, the next organelle that we are going to study are microtubules and microtubules organizing center. What is the meaning of tubules?
Like tubes.
Good. Okay. And micro means
Uh,
small, very tiny tubes. Now, you should know that this is a cell and there are different organelles in the cell. We have the nucleus, we have the chloroplast, we have the mitochondria, we have a large vacuole. How are they all in their places? There is a very invisible exoskeleton which is holding these organelles together. How are all your body parts intact? Ever wondered? How is your stomach in your place? Why is it not swimming around in your body? How is your kidney in its place? How is your lungs and hearts in their right place as you want them? Inside a skeleton. Humans have a skeleton, right? And all of our organs, all of our tissues are enclosed inside that skeleton. Got it?
yeah.
Same is the case with cells. Cells also have a skeleton. We call it the exoskeleton or the cytoskeleton. For example, we have a skeleton and inside we have all the organs connected and those organs can't move. I can't say that my heart is now in my stomach and my stomach is now in my place of the heart. That's not even possible because we have a skeleton that is holding it in place. Now, microtubules are the long, rigid hollow tubes found in the cytoplasm. They are very small, about 25 nanometer in diameter, and together with actin filaments and intermediate filaments, they make up the cytoskeleton. These actin and intermediate filaments will be discussed in chapter 15. But right now you should know that these microtubules are what? Long, rigid, hollow tubes found in the cytoplasm. They make up the exoskeleton like the diagram I have drawn over here, which is a very essential structural component of cells and helps to determine the shape. Microtubules are made up of a protein called tubulin, alpha tubulin, and beta tubulin. And these alpha tubulins and beta tubulins molecules combine to form dimers. Now, this is something very new and you have to listen very carefully. ये जो alpha and beta tubulins होते हैं, they join together to form dimers. And a lot of dimer means two. A lot of these dimers, when they meet together, they form long dimers. We call them protofilaments. This whole thing is known as a protofilaments. And when a lot of these protofilaments join together, they make a hollow tube. This hollow tube is basically known as the cytoskeleton. Got it?
Yeah.
I'll tell you again. Microtubules make up the cytoskeleton. One यहाँ बल्कि there's a picture over here. Look at this. ठीक है? What is this part called? Dimer. Got it?
Yeah.
Which is made up of one alpha, one beta. Now, a lot of these dimers join together. They make up what? Protofilaments. Got it?
Yeah.
Now, when a lot of these protofilaments join together, they are going to make a hollow tube. 13 protofilaments are going to join together to make one hollow tube. And this one hollow tube is known as what? Microtubule. Now you explain me the whole structure because this is a bit tough. Come on, you can do it.
So, dimers make up protofilaments, and 13 protofilaments make up one microtubule.
Exactly. And what are dimers made up of? Alpha and beta tubulin proteins. Okay.
Okay.
Good. Apart from their mechanical function of support, microtubules also have a number of other functions. For example, they can act as secretory vesicles and other organelles, and these components, cell organelles, can move. Imagine you're swimming and you are tied to a rope. When you're inside a swimming pool, a lot of people, a lot of, for example, the kids swimming pool, their floors are tied to ropes so they don't move from place to place. They can't go on this end, they can't go on this end. This is what an exoskeleton is like. Exoskeleton is the one that is going to make and move those organelles from one place to another. It's not like always the nucleus is swimming around in the whole cell. It's not possible. There is an invisible exoskeleton that is actually holding all of these organelles together. They form an intracellular transport system as well. Along these, microtubules, these transport can also be done. Then during nuclear division, they have a very, very important role that you will study in your AS chapter number 5, mitotic cell cycle. When we'll do chapter number 5, over here you will study that they have a very good role in nuclear division. Because what is cell division? Can you tell me?
Like mitosis and meiosis.
Good. So when one cell had some organelles, these microtubules basically help in cell division. How? These microtubules pull one organelle towards the other, and one microtubule pull the other organelle to one side so they can break apart into two. So they have a very, very important role that we'll see in chapter number 5. Microtubules form part of the structure called centrioles as well, which we're going to discuss our next organelle. And they also have these beating movements of cilia and flagella. The assembly of microtubules from tubulin molecules is controlled by special locations in the cells called microtubule organizing centers. Now, where is this whole assembly done? The one we have just seen over here, MTOCs, microtubule organization centers. Now, they are discussed further in the following section on centrioles. Centrioles are the ones where these microtubules actually arise from. What are centrioles? Centrioles are the organelles from which these microtubules arise from, and then they hold together these different organelles which are inside the cells. Got it?
Yeah.
So if you have to explain microtubules to someone, tell me, what are microtubules?
Um, they are tiny tubes in the cell that help form its exoskeleton and hold it in place.
Good. Okay. And what are their different functions? Any three functions, let's say?
So it helps in cell division. It holds the holds the organelles in place, and I don't know.
They're like a skeleton, holding organelles in place, moving organelles as well, help in transportation as well, help in creating vesicles as well, and they arise from centrosome. Got it?
Okay.
Good, good. These organelles are very tough. It's like 10 times tougher than IGCSE. Trust me, A levels, all subjects that you're going to study are going to be 10 times tougher, and you need 10 times practice as well. Self study from the, remember one thing, bio isn't tough because the only resource I tell all the students for biology is the book. You have never seen me teach biology from anywhere else. I don't use chemistry book that much. Okay, chemistry, I use a lot of PowerPoints. I'm going to use a lot of worksheets, a lot of topicals, a lot of other PDFs as well. But remember, book is the best source for biology. For biology, you don't have to read any other notes, any other PPTs. If you only focus on this one book, you can get an A star very, very easily. Remember that. Got it?
Yeah. Okay.
Now, all of these different microtubules, triplets of microtubules, form together one centriole. Okay. Now you can see this is one triplet of microtubules. This is one triplet of microtubules. These different nine triplets total, nine triplets of microtubules joined together to form one centriole. Now I'll explain the whole thing again. Alpha tubulin plus beta tubulin dimers created what? A dimer when they joined together. These dimers created what? Protofilaments. Got it?
Yeah.
Then these 13 photofilaments join together to make one microtubule. And now nine triplets of these microtubules, or you can say 27 microtubules, join together to form one centriole. And inside a cell, let's say this is a centriole. Centriole, this whole region is known as a centrosome. Centrosome is basically made up of two centrioles. This is whole, the centrosome plus the centriole, we call it as the microtubule organization center. So from these centrioles, these microtubules basically come out. Because obviously, all these, this is, look, this is a centriole. Out of these, microtubules are going to arise. Got it? These are microtubules. So they are present on the north pole and the south pole of your cells. Got it?
Yeah.
These centrioles are just present outside the nucleus. Why? Because they have a very, very important role in cell division. So technically, triplet of microtubules, one complete microtubule and two partial microtubules are formed. We call it a centriole. They lies closer and at right angles. Now, this is one centriole, like you can see over here. And the other centriole would be exactly 90-degree, lying always inside the cell. If you look at a cell in a microscope, this is a nucleus, and the centriole would be right over here, somewhere, let's say here. Two centrioles would be always pointing at 90-degree angle. And there's a very good specific reason for that, that we'll do in your cell division chapter. Now, this whole region is known as what? The centrosome. Centrioles and centrosome are absent from most of the plant cells. Why is that? Because plant cells divide differently than animal cells. Centrioles and centrosomes are needed for the cell division and for forming the exoskeleton and for providing extra structural support to animal cells. Plant cells already have a cell wall as well, number one. And number two, that plant cells divide differently. Their cell division, their mitotic cell division is quite different from that of animals. Your chapter number 5 is divided into two parts: cell division in animals, cell division in plants. A centriole is a hollow cylinder about 500 nanometer long, formed from a ring of short microtubules. Each centriole contains 9 triplets of microtubules, and until recently, it was believed that centrioles acted as MTOCs for the assembly of microtubules that make up the spindle. Now, out of these microtubules come out spindle fibers. Spindle fibers are the ones that are actually, kind of holding all those, pulling. They are very good at pulling. Okay? They are pulling organelles towards themselves. When during a cell division, this microtubule can be pulled back to this cell. These spindle fibers come out of these microtubules, and it is all done by this region known as the centrosome. They are also needed for the production of cilia, flagella, and all of that. So if I ask you, can you tell me what is a centriole?
So centrioles are made up of triplets of microtubules and help in cell division.
How do they help in cell division?
They like bring or like the organelles to one part of a cell when they want to divide it, like north and south.
Yes. ठीक है. And plus the region where we have two centrioles, what is that region called in this?
Centrosome.
Exactly. That is all you have to know. The rest will be done in chapter number 5. Is it clear till now?
Yeah,
Finding it difficult? Today's lecture?
a bit.
It's completely new. Believe me, it is new. I know that. Okay. We'll do a little bit, then we'll do some videos as well. Okay. So you can have a quick change of a study type as well. We'll watch a very 3D video. I have one that I'll show you. Ever heard of the term cilia and flagella? IGCSE you have.
Yeah, yeah.
What is cilia?
It's the like hair-like structures in the throat that beat mucus and dust particles.
Exactly.
And
Not
in the
exactly. Not exactly
yeah,
this party. That was the job of a ciliated cell. But exactly cilia is hairs. This cilia can be present anywhere. It's not compulsory. It's inside the nose. Both the ciliated cells are in your nasal cavity, the goblet cells and the ciliated cells. Cilia is hair. Flagellum is here as well. Flagella, for example, is the tail of prokaryotes, helping in its movement and all of that. So cilia, singular cilium, and flagella, singular flagellum, have identical structures. They are whip-like beating extensions of many eukaryotic cells, and each is surrounded by an extension of a cell surface membrane. They were given different names before these structures were discovered, and flagella are long and found usually one or two per cell. If you look at the structure, cilia and flagella are extremely complicated structures. They have 600 different types of polypeptides, and they all vary in how they beat. Some organisms, they are beating a lot depending on the different types of and the different number of that polypeptide. Now, cilia have two central microtubules and a ring of 9 microtubule doublets around the outside. This is referred as 9 plus 2 structure, and each MTD contains an A and B microtubule. Now, look at this. This is what a flagellum looks like. Okay? Now, this flagellum contains a basal body, which contains 9 triplets, and the cilium contains two more, let's say 11 triplets in the cilium. This is a cell, let's say. The flagellum starts something like this and then becomes thinner. Got it? So this part contains 9 triplets of microtubule. This part contains 9 plus 2. Got it?
yeah.
Good. The wall of A microtubule is a complete ring of 13 protofilaments, while and the B microtubule attached in an incomplete ring with only 10 protofilaments. Okay, you don't have to memorize all of these numbers. You just have an idea how they actually look like. The important questions we'll see at the end of the chapter, what do they ask? So figure 1.32 shows that each A microtubule has inner and outer arms. They are made up of protein dynein, and they connect with the B microtubules of the neighboring MTDs during beating. Now, how do they actually beat? Just understand what I'm going to explain, and that's all what you have to do. What are these protofilaments made up of? Dimers, And we are actually talking about like a lot of these, right? यहाँ पे सारे, these are all protofilaments. When they are going to beat, this dimer is going to vibrate that dimer, this dimer is going to vibrate the next one, and there's a whole motion that is going to take place. Like in atoms होते हैं ना metals में, how is heat transferred in metals? One atom to the other, they vibrate, right? So they are going to vibrate together, and one after one, they are all going to vibrate, and this causes the cilia to actually move like in a beating structure. Clear?
Yeah.
That's all you have to remember. Okay, don't read this whole detail. It's not in, no question is going to be asked in the paper regarding that. So the beating motion of cilia and flagella is caused by the dynein protein arms making contact and moving along with the neighboring microtubules. This produces the force needed for cilia to beat as neighboring MTDs slide past each other as well. Now, what are the functions? If the cell is attached to something so that it cannot move, fluid will move past the cell. And if the cell is not attached, the cell will swim through the fluid. अब इसका काम क्या होता है? When the cell is attached to something, fluid pass कर जाएगा. But these ciliated and these cilia are actually, the main function is them locomotion. Locomotion means movement. You are going to find a lot of videos on the internet, and you can see invertebrates beating cilia past epithelial cells में होते हैं, in airways as well, in nasal cavities as well. Goblet cells produce those mucus, dust particles, but those mucus you have to inhale as well. You have to take it in. That cilia is going to help move that, the mucus created full of dust particles along the stomach, where the mucus is going to be killed. Got it?
Yeah.
Okay.